I've seen so many UMAP plots over my career, and I've never seen anyone like really drive an argument from that. And like usually the plots that we make are like, you say this about reality and I say that, and you're like, no, look at my graph here. Like aspirin makes headache be less bad, and I'll be like, yeah, that graph is pretty convincing. I've never ever had that discussion with like anyone in my career so far.
Hello, everyone, and welcome to In Plain English, the podcast that is all about making science approachable, open source, and jargon-free. I am your host, Jamie Moffa, and today I am joined by three friends of the pod, Conrad Cording, William Lima, and Jake Walker. Welcome to the podcast, everyone.
Hey, good to be here.
And can you each kind of give a quick introduction of yourselves?
Wonderful. I'm Conrad Cording. I'm professor at the University of Pennsylvania. I do a lot of data analysis. I also deeply care about like making science accessible and good, and run an initiative called Community for Rigor, which tries to teach people how to do good experiments in data analysis. And I'm also a co-founder of Neuromatch Academy, which teaches neuroscience to lots of people.
Amazing. And Will, can you introduce yourself?
Yeah, hi, I'm William Lima. I've been this is my third time on the podcast. I don't feel very comfortable with the notion of being an expert guest. I want to be some kind of fuzzy expert guest. Okay, just, foreshadowing what's to come. But yeah, I have a PhD in category theory. I got my PhD in Cambridge. And I got interested in the UMAP algorithm because it's one of the big applications of category theory into like the real world, right? And I thought that was really interesting because I also dab a little bit in data science and stuff like that.
Great. And then last but not least, Jake.
Hi, my name is Jacob Walker. Too bad that you don't feel okay about being the expert guest, because I am certainly not going to be the expert guest on this one. But I've kind of known Jamie for a while. I previously worked in the labor movement, working with higher ed, specifically adjunct professors to unionize in the St. Louis area. And I have since retired from politics. I just sell houses now, which is fun and a lot easier than forming unions, even though it still isn't easy sometimes.
But without further ado, we're going to talk a little bit about a paper that made a big splash earlier this year, especially on science Twitter, but one that I think has a lot of implications for things that I care about with this podcast, like how we communicate science to other scientists and the public and how we do science rigorously. So the paper we're talking about is called Genomic Data in the All of Us Research Program. It was published in February of this year, and it had so many authors that they were just listed as the All of Us Research Program Genomics Investigators. I think there were over 50 authors in this one paper, which is a lot for a basic science kind of paper. And what the All of Us Research Program was trying to do is basically collect all sorts of health and genetic data from hundreds of thousands of people across the US, with the idea of getting a diverse sample to better understand health and genetic diversity in people who are not just white people, because a huge problem that we have in a lot of our existing data sets that people might use for understanding like genetics is that it's extremely heavily biased towards white people. It mostly includes white participants. So that was one of the main kind of ideas behind the All of Us research program, was to expand our catalog of genomic data that we have to pull from, so it's a little bit less of a monolith. And so this paper is part of that effort. They collected genomic data from hundreds of thousands, I think, of participants, and they set out to sort of like analyze some very basic features of the data. And the figure in this paper that we're going to mostly be talking about is figure 2. It's the figure is called Genetic Ancestry in All of Us. And what you see is a plot that has a bunch of like discrete groups of lots and lots and lots of different dots, kind of color coded by race in part A or ethnicity in part B. And what caused there to be an uproar on Twitter is that the different like groups of dots colored by race are pretty separate from one another. So what did they do to get this plot? Basically, they had all of this genomic data from thousands and thousands of participants. And when you collect genomic data, you have thousands of dimensions along which it might vary, thousands of different areas of the genome that were sequenced that might be different between each participant. So for each person, how do you represent potential similarities or differences across their entire genome? What they had to do, or what they did in this figure, was they used an algorithm called UMAP, which is what we're going to be talking about today, to condense it down into just 2 dimensions so they can display it on a 2-dimensional computer screen that is right in front of you. And when they flattened it all down to 2 dimensions, these discrete groupings of data were what they got. But as we're going to talk about today, there are features of the algorithm that they used, UMAP, that might mean that the underlying data is probably not quite so discrete as these different groups of dots that you see on your screen. So I'm kind of going to throw it to Conrad to talk a little bit more about how UMAP is used in scientific applications, and maybe kind of some of the like why you might use it, and maybe what are some pitfalls of it.
Yeah, I mean, like, the big problem that the scientists are trying to solve with this, is that the data that they have is incredibly big. We have, like, as you said, hundreds of thousands of data points. Let's say we make a matrix of that where that's the x-axis. So we have 100,000 on the right, on the x-axis, and then we have all the different genes on the other axis, which is also many, many thousands. So we have this incredibly big matrix. So I couldn't print that matrix and be like, hey, Jamie, look, here's how that works. That's not how it works. So they're basically faced with this. We have a really big data set and we want to show our audience something about the data. And like, that's kind of broadly the problem that they're trying to solve at that point of time. And this is a problem that we have in science all the time. Now we get really, really big data sets, and we want to show to people kind of what's in the data set. And that is what UMAP is often used for. So UMAP has multiple properties that make it super attractive for scientists, which is, even if you have a very large number of points, it's like really good to make sure those points fit into your image frame. There's lots of other ways of plotting the data where maybe you have a big clump of data points and then the rest of the world is filled with like occasional scattered outlier data points. In UMAP, like it tends to very nicely fit into the image. The second one is you want it to kind of like have structure. Like, I don't want to just show you the screen full of points and we don't see anything. UMAP is very good at making sure that what you see on screen kind of feels like it's meaningful. So, and in this case, it felt like super meaningful because not only do you see these things, they look like continents or something, islands if you want, mostly continents. They have like long bridges, these like elaborate forms. It's really beautiful. It's like profoundly beautiful. And then you have like one continent that sort of like these are Black participants, and another continent, mostly Black participants, and another continent where it's like, these are mostly white participants. And people then feel that by looking at these graphs, it tells us something about the underlying structure, that there's kind of like, like the way you and me interpret a map is not like, if A and B are far away from one another, they must be very different. Not like a Caribbean island is not like Greenland. They're from one another. They're like different in lots of ways. The big problem, and I'm sorry if I'm already giving that away, is that like at some level, UMAP is wonderful. It usually shows you structure that looks interesting, but it also produces a lot of structure out of more or less nothing. So what I'm saying already is like these islands, these continents I'm talking about, it's very difficult to interpret what they even mean. And in this case, kind of people are worried that that readers of the paper will interpret these continents wrongly.
Right. I mean, yeah, one of the biggest concerns was that, and this happens actually a lot, that a figure from, or not a lot, but like that a figure from a paper breaks out of the sort of scientific readership, which is what we want. We want like people who are not in science to know what we're doing. That's part of what this podcast is for. So a paper or a figure from a paper gets so big that it sort of breaks out of the scientific readership and into a broader audience and loses a lot of nuance and context. And there are definitely segments of the broader audience that would use the fact that these continents look so distinct from each other for racist reasons in this case.
Yeah, exactly. Let's push a little bit on that. Now, like the only thing that they have in their data set are Americans that they get the genomes from. Now, this figure looks like there's basically one continent for white folks like me and another continent for Black people, as if we don't live in the same space and like friends and lots of families and kind of all the things that like hold us together. And so that is, I think, why people reacted so viscerally to it. Now, like it's kind of like a map comes with like a promise that it shows us something about what's close to one another and what's different.
I mean, I could definitely see Twitter taking something like that and completely running with it in lots of different directions, very few of them actually helpful. So yeah, so I guess my question is, so the original project was to expand the data sets because we only had data historically for genetics on like white populations. So that was the point of this, was to make a bigger data set. And then someone decided to display that data like every race lives on a different island, and that's where things got.
But that's not someone who did it. No, that's the authors of this paper.
Oh, okay, I thought you were going to say it was you for a second. I was like, oh,
no. Conrad is the problem here.
I'm sorry. Let me be clear. I'm very happy to live on the same continent as everyone who's around me.
Me too. Say that.
Yeah. So this was one of the figures in this paper that was how the authors were displaying this data. And
got it.
The idea was to sort of expand the diversity of our data sets that we have for analyzing sort of trends in genetics, and with the idea that like there's this whole idea of personalized medicine, or like medicine basically that's going to look at like hyper specific factors to you, like your like particular mutations you might have in a specific gene that would affect how you're going to respond to a particular drug, things like that. And we use large data sets like this one to kind of understand where people fit into like different broad like genetic populations, to understand like, oh, you know, maybe this person would respond better to this than this, right? It's sort of becoming a new thing. It's interesting because it's like big data and personalized medicine together. So like the very, very, very micro of like, how are you going to respond to this drug? And the very, very, very macro of like, we're going to understand that by understanding how you fit into this like big structure of data.
So what sounds like is interesting about it is this wasn't necessarily created just to explore difference in race and things like that. It just needed a more diverse data set to see just more variations generally, but previously race has been one of the variations that wasn't included.
Yeah. Well, and I think they wanted to show off. I mean, this is a scientific tour de force. This is a like big undertaking. And so they wanted to show off, and they wanted to show off that they had in fact collected data from a lot of previously very underrepresented groups in research. So like, I think the underlying motivation behind it was quite good, but sort of the way that it got interpreted the way that the execution happened and got interpreted was not as good.
Yeah, so look, I mean, that is the weird thing then. Like, the real thing that they did for science is an incredibly valuable service, which is data from so many people. This is something that's going to be transformative for all kinds of medical research. Like, it has like hundreds of thousands of people in that data set from all kinds of origins. It's like really incredibly broad, and it powers science. But it's not science. Now, let's be clear about it. This is not a paper that tries to understand something about the world. This is a paper that just says, we produced this unbelievably cool data set. Let's go. But they felt compelled to put something into the paper that sort of says, like, look, and here we have like some insight. And that insight arguably is misleading.
I see, especially if it gets out of the realm of people that kind of understand what the context was before and why it was created.
But even like within communicating to other scientists, because it's potentially, and like none of us have access to the underlying data set. We don't know, but it's potentially like creating these islands of people where like in the actual data that distinction doesn't really exist. Conrad actually made a cool little app that I'll link on the website, but generally what it does is it takes this like uniform scatter plot of points, that Conrad's app, right? Not all UMAP, but takes this uniform scatter plot of points, and you can apply the UMAP algorithm to it under different parameters that you set by sliding numbers around. And depending on the parameters you set, it can take this very uniform scatter plot of points and turn it into something that looks like there's distinct categories inside of it.
Right. No, so let's keep going with this island analogy. So we start with just like a continent that it's two-dimensional and it has constant density of people. Basically, like every 100 meters, there's a person. And now we take UMAP map and apply it to this. And UMAP, like a lot of algorithms, has underlying so-called hyperparameters. So basically, it has two dials that we can rotate. Now, if I put this one island where everyone definitely lives on the same island, per definition, because that's how we design things, we put like all this data into UMAP, and I turn those knots. I can make it so that it looks like there's 4 continents that are entirely unconnected. Or I can make it that there's just one of them. Or I can make it that there's like little streets and all the rest of so basically, by changing the hyperparameters, I can more or less choose how the results will look like. That means that if we live on a continuum, or if you want, like, it's not that like there's 2 kinds of humans in our data set. It's really just like everything exists, and any mixture at the output of the algorithm, it looks like there's islands. And that statement that there's islands, we could now start saying like stupid things about these islands. Maybe people on like that island full of like German people, and maybe Germans are like in the southeast of that like original thing. They all end up being on one island. And then it's like, okay, well, those are the Germans. Let's not talk with them. They're not funny. So but in reality, there are like people that are more German and less German and more funny and less funny. And like on German island, there's plenty of people who are like very funny and plenty of people like me who are not very funny. And kind of like if we use these algorithms, they make us wrongly kind of believe in there being discrete structure, that like we live on islands in a world where like things are very continuous.
Yeah, if I could just come in and say one thing that I noticed in reading the paper and looking at the talks is that there's a lot of kind of bit of a confirmation bias. Basically, they show that the algorithm is very good because it outperforms other algorithms in separating things which we know belong in different clusters, right? They show, for instance, like, oh, look, it can recognize digits, the digits from 0 to 9. Here's another algorithm. Here are the clusters that it produces, and UMAP produces even clearer clusters, right? Or other stuff like that, that basically, they're basically worried about the sort of type 2 errors, right? They're worried about like, it's when there are patterns, it's catching them and it's doing it better and faster than the other algorithms. But the other question is, but what if there aren't patterns? Will it show us patterns if there aren't any, right? And that's the thing that is problematic, right? Sometimes it's just, there are no patterns at all, but you start twisting the knobs and suddenly something appears and you go like, yay, can I get a grant, please?
Yeah, for real. I mean, there is definitely from the research end an incentive to make your pretty UMAP graph look like it's, look, I have these discrete clusters, like, maybe you're mapping out the genetics of, like, I'm a mouse researcher. So you're mapping out something about mouse genetics, and that you have 2 clusters of mice, and you're like incentivized to make it really, really look like distinct that you have like these 2 different groups of mice. But when applied to real world data, that can get a lot messier.
I mean, what jumps to mind for me going to like a different department on the college campus, because it's how I'm how my thing. In geography, mapping, the world's like spherical surface onto a 2-dimensional plane. I mean, you lose a lot of data in doing that, and then the same thing that people talk about with the different projections, like the initial projections that we used, like the Mercator projection is crazy. Like it makes like Greenland and Europe look gigantic. So and like the general like edges of the map. So yeah, I definitely see how there's like kind of a well-worn path here of how someone would see that and then say, like we've seen this before of the way that you display data, especially like when you're trying to simplify it and taking out some of the complexity, because like you can't, like Conrad was saying, you can't just give all this complexity to people. There's no way to transfer that much information visually. But then we have to be very mindful of what we take out and what we leave in and the things that we don't show. It's very interesting.
Will I wanted to ask in a way that we're going to understand, because this is super complicated. What is it about this algorithm? Like, why does it do that?
What is it doing? Okay, here we go. Buckle up. I like that the map projections was brought up, because that's a really good analogy, right? Getting something that lives in 3D space and projecting it into 2D, right? And because that's what you're doing, like, because UMAP is basically a dimension reduction algorithm, right? It's trying to reduce the dimension of your data, right? Very similar to like when we reduce a sphere that is a 3-dimensional object into just 2 dimensions, right? The thing with these map projections, right, is that they all have a formula, right? They all have a formula that we know exactly what is going on, right? So for instance, the Mercator projection, we kind of know what's being distorted, but we know what's being preserved. The Mercator projection preserves angles, right? It's a conformal map, right? So if you're navigating and you need to know get the angles right, but the distances don't matter that much, Mercator is fine. But then there are others which preserve area, but then the angles get all kind of wonky, right? So there's always this thing of when you're projecting, you got to keep in mind, what are you losing and what are you preserving, right? That's one thing that's important, right? Because as far as dimensionality reduction goes, right, there's 2 families of algorithms. Like there's the matrix factorization algorithms, which are like principal component analysis, right, which are very old and very fast. Those guys are super fast. And then there are neighbor graphs, right, where you're trying to do basically look at points and find their nearest neighbors and try to construct graphs of those. Like one famous one is TSNE, right, which came out in 2002, I think. No, I think it was just SNE at the time, and then TSNE came out a bit later. And UMAP is one of the more recent ones, which is, again, a neighbor graph, but is super fast, right? It's much faster than TSNE, which is kind of like the state-of-the-art one, right? And what it does, right, is it constructs a graph of your data, because your data is just like a bunch of isolated points. It's going to construct a graph, right? And the edges of those graphs represent neighbors that each point has, right? So each point, you're going to connect it to its neighbors, right? But this is going to live in a very high dimensional space. Once you've done that, you're going to try to project it onto a lower dimensional space trying to preserve some things, right? Just like when you do a projection of a sphere down into a flat map, you're trying to preserve some things, right? What UMAP does really well is construct this neighbor graph in the high dimensional space. It constructs it in a way that's really different from all the other algorithms, right? And that relies on a lot of heavy math, a lot of math that's really hard to understand, right? So basically, like, how would we go about just constructing a neighbor graph? Because we're trying to get clusters, right? Our data might have clusters. And if it has clusters, we want to preserve those clusters, right? So you want to find out for each data point, who are the guys who are next to that data point, right? And then maybe there's another cluster somewhere else, and you want to find out, given a point there, who are the neighbors of those guys, right? You can think of it kind of like if you live in a city, right? And you want to connect yourself to your actual neighbors, the people who live right next door to you. How would you go about that, right? Well, you start with yourself, and then you maybe arbitrarily construct a perimeter radius. You're going to go like, no, everyone who is my neighbor lives within 100 feet from me, right? And then you say, like, all those guys who live from 100 feet from me are my neighbors. And so I'm going to join, myself to those points with edges on my graph, right? But what if you live in a much less densely populated place, right? Where your neighbors are really far away. Like, for instance, right now I live in St. Louis, and my neighbors are probably, 30 feet away from me. But I lived for 5 years, I lived in Alaska. And so a lot less densely populated. My neighbors were a lot further away from me, right? So there's like two issues, right? So first is, how do I determine this perimeter, this radius in which everyone who's inside is my neighbor? And the problem of this radius being arbitrary, right? And UMAP addresses those two issues, right? The first one is that it uses different units of measurement based on how densely populated your clusters are. So for instance, if I have my house in Alaska, right? My neighbors are 100 feet away from me, right? And those are the people who are considered to be my neighbors, right? But if I live in Japan, right, my neighbors are maybe 10 feet away from me or something like that, But this creates a problem of compatibility, right? Because when I lived in Alaska, all my neighbors were really far away from me, but there was one lot next to my house that got bought by someone, and they built 6 houses in that lot, 6 tiny houses there, right? So now there's this really densely populated cluster there right next to a bunch of sort of like spaced out things, right? And so what happens is my criteria for being a neighbor is incompatible with their criteria, right? Like the people living in that lot, they all think their neighbors are just 20 feet away, while I think they're all 100 feet away, right? So I consider them to be my neighbors, but they don't consider me to be their neighbors, right? Because these clusters have different metrics, right? And UMAP kind of solves this problem, right? And it also solves the problem of, well, what is the threshold of where I cut off who is my neighbor and who isn't, right? Isn't that a bit arbitrary? It also solves that problem as well. So this notion of the cutoff being arbitrary, it does it by not having a fixed border and having a fuzzy border, right? So now we're not saying like either you are my neighbor or you're not. We're calculating a probability of you being my neighbor. The further away from me you are, the lower the probability. The closer you are to me, the higher the probability of you being my neighbor, right? And the incompatibility, this, of like, we're using different metrics here to measure things, right? Different units of measurement almost, right? This way of sort of ironing out incompatibility is where the category theory comes in the form of adjunction theorems, right? Which are basically approximation theorems, right? And so basically there is a way using this powerful machinery of category theory of using adjunctions to kind of iron out these incompatibilities and basically find a graph that is where everything is compatible, that is a sufficiently good approximation of your original incompatible graph, right? Because the original graph might have two edges between each node, where the way they measure distance and the way I measure distance between us is different. And then these adjunctions, they basically iron this stuff out, right? And I feel, does that make sense? Do people have questions?
Yeah, let me ask for like an intuition builder, which like imagine like the real data would live on a two-dimensional plane. And just like you explained, there's areas with very low density and areas where lots of people live close to one another. Is one way of meaningfully thinking about it that like first order approximation kind of takes these people that are close to one another, like moves them away from one another, from their neighbors, and takes people that are very far away from everyone and moves them closer to everyone, so that it's like more producing like a uniform map of sorts.
Yeah, exactly. That's where the U in UMAP comes from, right? It stands for uniform, right? UMAP is uniform manifold approximation projection, right? The way it constructs this distance, these units, is it looks at who is the closest neighbor to you, and that will be your unit. That will be your measuring unit, right? So if they're far away, your measuring unit is 100 feet. Right. But then if you go to the really densely populated area of your graph, if it's just 10 feet, that is your measuring unit. Right. And so you're secretly using different units, but locally, everything kind of looks the same, but you're using different units that you can observe from afar. But in each context, everything seems very uniform. It seems like, oh, yeah, I just have like 3 guys close to me because they're all inside the 3-unit radius, right? Or 5-unit radius. And same as the other one. Oh, yeah, I just have 3 or 4 guys next to me because they're all inside the unit radius, but the radius is much longer. So it seems uniform, but you're kind of cheating. You're changing the units, right?
Yeah. And so let's maybe push a little bit on the uniformity. This is amazing if you think about what we want to do with a figure. Like in the figure, we have like one window, like where all data points need to appear. The fact that they kind of become roughly uniform means that it's much easier for me to basically take a photograph. Now, if I take a photograph of the group, I want that every face is basically the same size. So UMAP does something like that, of like basically making sure that everyone's the same size, which is useful. But can you explain the second part? Like the problem is if we're in many dimensions, say 3 dimensions, imagine we all lived in 3 dimensions. UMAP still puts them onto 2 dimensions. Can you give like the listeners a little bit of an intuition? Like if I have like the Earth, and it's filled with stuff, how can we make that Earth that's filled with stuff kind of appear on just one map?
Right. Yeah. So again, like with the way they did it with the Earth would be like using a formula, right? But now we're using like more machine learning techniques, right? You have your high dimensional data set, right? You want to find a low dimension approximation, right? You just basically go by trial and error, right? You just put a bunch of points down there, try to construct a graph the same way, and then you measure how similar these 2 guys are. And if they're not very similar, you move the points around a little bit more, try to fix it here and there, and then measure it again. And like, oh, is it looking more similar according to some kind of way of measuring, right? Because like I said, there is no formula, right? It's just this trial and error, which has the fancy name of gradient descent, right? You're basically trying to adjust things until you find it. No. We keep moving this a little bit and it doesn't get any better. Maybe this is as good as it gets.
So, but to be clear, if we want to take the whole content of the Earth sphere and represent it as like a 2D surface, we have no choice but kind of having cuts in it. Like we need to kind of first have some parts that are like outside of the world, and then there's going to be a piece of the inside and parts of the outside.
Yeah. So this is one point where t-SNE and UMAP are kind of different. And I'm not quite sure why they chose different loss functions, right? So T-SNE, right, which was the much older algorithm, what it does is, look, I want things which are close together to remain close together, and just make sure that nothing that is far away comes too close, right? Like if something is really far away, don't let it get too close, right? So that's why it spreads stuff out all over the place. UMAP uses a different loss function. It uses a cross entropy function, which is like when it measures this, there's a component there that is measuring other things which are close together, sufficiently close together. Yeah, they're good. And then there's another component that's going like, but what about the things that are far away? Are they still far away? Are the gaps still being preserved? Oh, yeah, the gaps are still being preserved. And so that's good, right? And if these two things don't get preserved, the gaps and the clumps, then it starts going like, no, this is a bad approximation, So that's kind of the loss function that it's using there, right? To compare these two versions of the graph.
I think I'm understanding, but I was going to use an analogy from real estate to see if I am understanding. So, I mean, say, one is selling a condo and is responsible for putting a price on that condo. So yes, like the world, and it's in the Central West End. If we use, there's two people here from St. Louis, at least. The Central West End is a very urban, very dense neighborhood. So like the best data that you're going to get on what to set that price for would be the other units in that same building that goes like vertically, right? Because it's the closest together. But if there's a penthouse at the top of that building, and your unit that you're selling, unfortunately, is not the penthouse, it's the the middle of the road condo in the building, then you probably wouldn't like care that much about the penthouse. Additionally, if it were a single family house in that neighborhood, then you could go further away to look at similarities to try to set a price. But if you have all of this data from just the building itself, then you should use that data in the same way of like, if you had a place in a rural area, your data just may not be as good because you could go into a different county with different taxes or something like that to find your next comparable property.
Yeah.
Is that kind of, so I almost kind of feel like what you're saying, William, is kind of what we do in our brains as real estate agents when we look at a house, because there's like, if algorithms could set, prices for houses, Zillow wouldn't have lost like $1 billion buying houses 2 years ago. So that's kind of what's coming to mind is like when I'm looking at different comps, there's a lot of art to it where you're putting this out, you're putting that out, you're taking this data point in, putting this data point in, using like common sense or your experience as a professional. But that I kind of what you're saying is like all these algorithms are trying to kind of replicate that on a large scale and be able to multiply like many thousands of times to make sense of like a lot of data.
Yeah, exactly. And then the thing is, a priori, they don't understand that things suddenly become different, right? That's the problem, right? You give it like one criterion, right? And then it tries to do that globally, right? And then, well, that doesn't work because, one area is more dense than the other. It needs to be able to make the switch. No, this is less dense, this is more dense, fine. But then when they start getting close to each other, it doesn't know how, right? It doesn't know when to make that switch, right? And that's when where the category theory comes in, that it finds a way to average out to make this smooth transition so that things are kind of compatible. Things that would be incompatible at first become compatible. And that is like one big black box, right? That it's kind of like, how exactly is it doing this? It's using one big adjunction theorem to sort of average these things out to make these transitions smooth, to make everything kind of compatible,
Yeah, so I kind of wanted to pull us a little bit out from the math weeds, and what you said about it being a black box is actually kind of the first thing that I thought when I saw everybody talking about it on Twitter and started like trying to figure out what UMAP was and realized like, oh, most people who are using this, like most scientists who are using this don't know how it works, which is not like always a gigantic problem because you can't understand perfectly how everything works. Otherwise, you'd need to get a PhD in like 5 different subjects. That could become a problem for researchers applying UMAP to their data and then communicating what they find to other researchers and to the public. So I kind of wanted to talk about like how does that create problems, and then like maybe how can we mitigate those problems?
I mean, here's where I see the real problems with it. If we zoom out, it's a method that makes us a map. We all are used to reading maps. That's why maps are so attractive to us. We know how to read maps. What does that mean? It means that if points are close to one another in the map, they are close to one another in the real world. And if points are far away on a map, they're far away from one another in the real world. That's like what we use our map reading for. Neither of which are, of course, true for UMAP. Fast order is trying to do something like that. If it can, it tries to have nearby points be close to one another. But if we're in high dimensional spaces, you and me, we can be different in so many, many different ways. How could all these ways, how any pair of us is different, map onto one space? Well, it can't. And so therefore, if you want the typical UMAP 2D surface that we see in these genomic studies, explain a tiny part of the overall variance. So data points can be close and in the UMAP, and they're very different, and they can be very far away, and they're very similar.
Yeah. So I guess maybe even what do researchers even use UMAP for? I know we started out with that question, but it seems to me that you can't really analyze it because it's doing all these black box transformations on the data. You can't really even use the distance between points as any kind of metric of how different they are because of all the stuff we've just been talking about, that the distance might be the distance is different because of what Will was saying about the neighbors. And sometimes neighbors are really close together. Sometimes they're farther apart. And UMAP is taking all of that and making it look uniform, which looks pretty in a graph, but you don't really actually know how similar or different those points are in your actual data set. So how can it be used as a part of doing rigorous science?
Yeah, I don't know. I think we've already seen how, sometimes the hyperparameter tuning can be so fragile as well that, you just tune it to one way and then suddenly your data gets completely split just because of some idiosyncrasy of the data as well, right? And so there's that on top as well, is that eventually you're kind of p-hacking it a little bit, right? Twisting the stuff until you finally get something that stands out, and you're not quite sure why. It's just some idiosyncrasy of the data there that just happens that when you set because one of the parameters is how many neighbors do you expect each point to have, right? If you start toggling that, eventually some weird stuff happens, right? Some discontinuities happen, right? And you're not quite sure why. So yeah.
Right. When I was playing around with Conrad's app, if you kind of had a medium amount of neighbors and made them all be the other knob is sort of how far apart they're clustered. The other one is minimum distance. So is that unit of your neighbors are 10 feet away from you or 100 feet away from you that you were talking about earlier, Will?
I believe so, yeah.
Okay. So if you have a very small minimum distance, meaning that you're only looking at the points that are the absolute closest to each point, and you have kind of a medium number of neighbors, that sort of seems like the sweet spot, at least in the Conrad's app, for creating clumps in the data where none really exist. You're kind of saying we can have some amount of neighbors, but they all got to be really close to you. So it's going to segregate, even though the data is uniform, it's going to say that this clump goes over here, that clump goes over there, because it doesn't care again about really how far apart those points are in real life or how different they are. It just wants to take the parameters you gave it and make a graph that's going to fit those parameters and be as similar as possible to the original data. But if the parameters don't allow it to be that similar, then it's going to create these clumps. Is kind of my understanding.
So I asked on Twitter today kind of what UMAP is useful for. And it sounds like the consensus at this point of time that it's a method that gets us started on something, a method that gets us to have some insights on what's going on, but no one is really good at like saying what that insight is. It sounds like more like people like looking at these maps and I think the vast majority of cases that I use are actually in papers of a kind that I think we shouldn't be so negative about. Like take this paper, the All of Us study. They provide amazing data that is going to be used by thousands of groups to ask important questions about health. They just use this algorithm to make a pretty figure that kind of everyone in the room knows means pretty much nothing. And the majority of these figures that I see are of that kind, where people are just like, look, we produce a great data set with lots of stuff. And also here's the UMAP. People are like, that's a pretty UMAP, but it doesn't strike me as being part of science. It's a part of a process that makes a really appealing figure that shows all the data points. That is how I now think about it.
Yeah, I'm just toying around here with your app because it's so friggin' entertaining, right? And I just changed the minimum distance from 0.4 to 0.42, and what looked like two clusters just joined into one. Do you get me?
That's right, yeah.
So it would be interesting to have an animation, right, of continuously varying the hyperparameters and see how that changes,
Yeah. No, that would be very nice, because if you do it continuously, you will find that there's like probably some periods where things very smoothly change, and then periods where it like jumps and really changes the interpretation. The slight problem is not like the method is probabilistic in and itself. Therefore, you'd need to somehow like freeze the random seeds, but you can do that.
Yeah.
So my concern is like Conrad, it sounds like your point is that mostly people are using this in very exploratory papers about large data sets that they don't know a lot about yet. And, eventually people are going to do a ton of research on this and like much more rigorous research with much more like statistically rigorous like computation methods on maybe smaller data sets so you can understand features of like a subset of the data set. I guess so it's good in that people aren't really using UMAP necessarily as part of like a paper that's hypothesis driven and answering a specific question about the specific data set. I guess my concern is that if someone doesn't know that and they look at the All of Us paper and they look at this UMAP and they see, for example, it's kind of created these two clusters out of the dots that are labeled as like Asian. And so now I'm going to form an entire research hypothesis about the genetic differences between like different populations of Asian people, where those two clusters might not actually represent a real feature of different populations of people from Asia, right? Like, I'm concerned that this can be used to generate hypotheses that are not grounded in reality.
Yeah, we will see clusters that aren't there in reality. And that is always problematic.
It kind of, so you're saying basically like the UMAP stuff, it doesn't, so it's not useful for, zeroing in on these research questions. But I mean, wouldn't it be useful for deciding which data set to use, possibly, if you're trying to look at one thing or the other? Wouldn't it be, back to the real estate thing, like, do I want to focus my business on 2-bedroom houses or 3-bedroom houses in a certain market? And then you can get like an overall view of like what that would be, or you know what I mean? Like that, is that something that it could be used? Because don't you have to choose a data set before you start doing research?
But Jake, if that's what you want to do, look for 2-bedroom houses and look for 3-bedroom houses and plot what the differences are. So I don't really see, and apart from very few exceptions, how this really guides that choice. And I wanted to briefly come back to Jamie's points. Like, are there worries of non-rigorous things going on? Like, people view it as just, it doesn't matter, it's just like a pretty picture. But there's parameters that I can turn. And there are things that people will find very cool, and things that are that people will find less cool. Like if you, in many cases, you have these extra features. Maybe you have patients who have a disease, patients that are healthy, young people versus old. You have these classes that are in there. And people want to say that their data is indicative about that. So I'm very concerned that they basically tinker with things until the graph looks amazing. And amazing in the sense that there's basically an island where everyone on that island has the disease, and another island where no one has a disease. But given that it produces a lot of structure, even if there's very little in reality, there's a big open possibility that people will basically give us those graphs that suggest things that aren't true, that they basically comes from them tinkering with data.
I wonder if, I'm thinking here, what would be a convincing UMAP argument? So if maybe someone showed you, that no matter how you tinker with the hyperparameters, you always get two clusters, right? Hey, I've toggled the hyperparameters all over the place and everything, and there's always two clusters here. Maybe that would be a convincing argument, maybe?
Yeah, but that's never... as far as I know, that's not what's ever happening there.
Yeah.
No, look, here's the problem. In reality, data like genetic data has lots of clusters. People come from countries. People are the first generation of people from one country having a family with people from another country. So it's not just that you have a small number of clusters, but you have clusters and clusters that are mixtures of clusters, and you don't have manifolds at all. This officially UMAP is a method that is meant to you have a continuous manifold, but yeah, right. In genetics, you have continuous manifolds. Give me a break. You don't have continuous manifolds.
What is it? What is a continuous manifold? I actually don't know what that is.
A continuous manifold is, imagine there's two aspects in the world. What do I know? Maybe tall people versus small people, and I plot the height of everyone. It's going to be a nice smooth distribution with a peak in the middle, and outliers on both sides. Not we have the occasional basketball athletes and we have the occasional person who's very short, but it's a continuous distribution. That is, and those continuous distributions, they're usually what people understand about manifolds. Take images. Now you can say, I show you photographs of two different objects, maybe a bicycle and a car, and I show you all these, and all these images can be small or big. They can be rare, can be brighter versus darker. There's kind of a manifold of objects that we are talking about here. Two manifolds in that case. Now, the manifold of all bikes, the manifold of all cars. In that case, the methods that we have from UMAP are really good, because you can say, if you give me enough data from enough bicycles, can kind of tell you about what the dimensions are along which bicycles vary. The problem is bicycles are still discrete, not there's bicycles with and without shock absorbers and things like that. But imagine it was just in terms of size and height and things like that. It lives in a manifold, and these manifold algorithms feel like they're doing the right thing. It's just that we now use these algorithms that are meant to do one thing, which is figure out how these manifolds lie and apply them to things where we don't even expect that manifolds should be a thing, just because they make pretty pictures.
Okay, I might have missed this, but what is a manifold? Besides I don't know what it is on a car or...
Yeah, let's sketch a case where things would work extremely well, and let's sketch a case where things would work very, very poorly. Imagine there were basically two types of genes, let's say A and B. And all our genes would be A, B, 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 A, and B, 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 B. And it basically, the way all of us would vary only along this one axis, how many A's we have versus how many B's we have. Then you can say, UMAP is wonderful because if you want, like 17 A's is the neighbor to 16 A's, which is the neighbor to 15 A's, and so forth. And we can figure out everything that we have there. The problem is genetically, we live in a very, very high dimensional space. And we don't have continuous distributions there. Like it's kind of like you either carry a gene or you don't carry it. And with it comes all kinds of things. Or you grew up in some country, and you have like all kinds of genes that kind of come by virtue of me growing up by me like basically having German heritage. And so in that case, it's rather that there's kind of people from one country and people from another country, and within each country there's a distribution, but there might at first like not be like a simple overlay in between. So it's not that we have like all possible things happening in between, and all possible things don't work in high dimensional data sets in the first place, because kind of like the space is too big to like even in a way have all possible neighbors.
Yeah, this is one thing that's kind of bizarre about math, is that so many of the powerful theorems depend on things being continuous, right? Depend on continuity and everything that's really well behaved when you have a continuous spectrum, right? And when you don't have that, you don't have the theorems, everything's ugly, and you can't use, a bunch of tools that you have. So people usually assume that everything's varies continuously because it fits the math, right? It makes the math easier. Such as is the case of UMAP, right? Let's assume that all our points are in a manifold, that everything is varying smoothly and continuously. Let's just assume that. It's a pretty heavy assumption, right?
So it would kind of be the difference between like a car manufacturer where there's 10 different car manufacturers and they all have variations that are kind of, you can find all of them, right? Whereas like comparing that to like a bunch of home bakers who are making sourdough loaves at home and then trying to plot car points and then like sourdough loaves. Like there's going to be so many variables. You're not going to really know how each variable
interacts.
Yeah. Maybe kind of like these bespoke things rather than something that's like mass produced. And then the cars would be in a manifold. Is that the correct way to use that term? And then the loaves or the people or whatever, the irregular would not be in a regular manifold. Is that the correct way to use the word?
Okay. Like if we want to push on the word manifold, like, well, do you want to give the mathematical definition of manifold? Should I?
Sure. Why not?
Oh no.
I mean, Jake, you brought it upon yourself.
I did. I asked for this.
The motivation for manifold was just a geometric object, right? A sphere is a manifold, okay? A sphere is a smooth manifold, right? Some wacky thing like a torus, a donut, is a manifold as well. It's a smooth mathematical object, right?
Okay.
The interesting thing about manifolds is that when imagine if you have a really big donut the size of a planet, right? A donut the size of a planet. If you're standing on it, you're gonna think the same thing that some people sadly nowadays believe, is that it's flat. Is that when you're there, because it's so big, it looks kind of flat, just like the Earth kind of looks flat if you don't know any math, right? Right, and physics, and let's not get into that. But anyways, so manifolds locally, they look flat in 2D, but globally they might have other kind of shapes, And so that's the thing. But usually, it's a fancy word for geometric object. A smooth manifold is a smooth geometric object. Continuous manifold is one that doesn't break into bits. It's all connected, continuously connected. Differentiable manifold, and stuff like that. You have something to say, Conrad?
Yeah, like basically the key thing, and there you see how it relates to UMAP, is on a manifold, I can always walk in some direction. No, and as I walk over my manifold, whatever that manifold is, I would be walking through the 2D space that UMAP usually does. So you need a space in which you can walk.
So if something isn't, so it would be almost like the map analogy, like you need continuous land and not something like off in the distance, like a fractal or something like that would be off. I see. Yeah.
That's right. No, I have blue hair and you have brown hair. And there just is nothing in between. You can't walk from blue hair to like your hair color, the way I think about it. At least not within the set of hair colors that humans actually carry. They usually either really blue or they're like really brown. There's like very little in between. And therefore, if you did UMAP on us, kind of, you would on our hair colors, you wouldn't
be very useful.
So, Conrad, are you saying that you genetically have blue hair?
Well, my daughter has the same shade of blue, so it seems to be that it's genetically inherited. Yes,
good enough for me.
Backing up, these plots are pretty for scientists to look at. And good for demonstrating, like, we had this huge data set and we did something with it. But like, here's just sort of a pretty representation of our huge data set that like later people are going to actually investigate and have like all sorts of like rigorous questions that they ask about it. But like, since we like looking at these pretty maps of our data so much, we're going to keep making them and we're going to keep putting them in papers. And those papers are still going to be read by people who are maybe not scientists, or if they are scientists, maybe don't necessarily know the intricacies of the like type of science that's being done, or, you know, don't know that a UMAP is a pretty graph to show the largeness of your data set and that you've read sufficient papers to know what UMAP is. So basically my question is like, if we're going to keep doing this, what are some ways that we can maybe mitigate misuse or misinterpretation of things like UMAP by the public who we hope read our papers, who we hope like actually find out what is going on in science?
I mean, like, I think I can give a recommendation there. It's almost like all UMAP graphs should come with a warning. This is a map that does not well approximate reality. And we should be super careful at the follow-up studies. You know, like, I've seen people do the wildest things with UMAPs. They like take UMAP and then they take the output of UMAP and cluster it. That makes no sense because UMAP makes the clusters. Basically,
you already clustered.
So what do? They UMAP it twice?
No, they take UMAP and then they run k-means on the results of UMAP.
Yeah, man. I mean, here's the thing. Looking at the math as a pure mathematician, I'm like, wow, this is so pretty. This is so beautiful, right? It's so interesting and everything. But I'm highly skeptical, super highly skeptical when people try to use mathematics to give this air of precision, the mathematical precision. You know, where in any sphere, especially, this is going beyond this chapter here, but especially in economics, you know, I'm very skeptical of that branch of like econometrics and say like, oh no, this models perfectly, you know. But so I don't know, man. I look at the stuff and I go like, this could mean nothing. This could mean absolutely nothing. You know, that's my 2 cents. You know.
That's right, and it's very likely that it often means absolutely nothing, and it is likely that scientists misinterpret it, and I think yes, there should be a warning label.
So we're meant to tell people that this thing that we did that looks very pretty is in fact like not really useful for anybody.
That's how I view it.
Is there any sort of like, because I think William was talking about grants and stuff, and like, so is there any acknowledgement that like within science you have to do marketing to be able to like get like could that be the application for it? But then also I guess it's misleading, so you know, then I guess it's not like a fair thing. I just think about it as, you know, there's sometimes like visual aids you can use for advertising whatever that may not be the most accurate per se, but gets the point across that you're a really good agent. You sell all your houses for lots of money, you know. So, like, that's
you have a very doctored graph of like, these are all the houses I sold, and look, they're all like, you know, $400,000+ Like,
I'm looking up UMAP right now and seeing how I can put all my transactions on it. So
But I think it's exactly that. Like from a marketing perspective, if you want to publish your data papers in top journals, they expect you to have the following. They expect you to have a fast figure that basically just says, this is an unbelievably big data set with lots of cool things in it, which is exactly what all of us folks do. And then at the end of that figure, there's often like results and methods, the figures in between. But at the end, there is like the teaser of science. We're doing like a little bit that just justifies why we would be publishing this in a scientific journal. And the UMAP thing kind of like tends to, from a salesmanship perspective, fill that role. And we did UMAP, and like, look, we have like on UMAP this thing looks like a cluster, and it's all people who have the disease, or it's all people who are old, or it's all people, or it's all neurons with this or that property. And in that sense, it kind of pretends something that the papers don't actually deliver on.
Okay.
Yeah, I'm really scratching my head to try to find something that would be a legitimate use, right? I wonder if, for instance, if you have a very large data set, right, you run UMAP on it, and then you say that, no, but I found a really good method to do data cleaning on this data set. And now with the same parameters, now we see that the clusters become clearer or something like that. I don't know. You know, instead of using it as some kind of other threshold for something, you know, as a benchmark for something, I don't know. Because as just like trying to sell a picture of reality, you know, it seems very problematic, you know.
Yeah, as I try to think through like applications for it, it all comes back to like the only applications, I guess, from what I'm hearing, are like those that may potentially mislead people, which is generally, I think, as science, like with you guys being scientists, not something hopefully you'd want to do. There are some out there, I think, that want to do that, but
I gotta say, the category theory community loved it when UMAP came out. They're like, wow, we're gonna get so many grants now, finally. You know, yeah, we're really doing like a lot of mental acrobatics here to try to find, you know, I like how it shows how benevolent we are, you know, like, no, let's give it a chance.
Yeah. So something else that I was talking to another friend about this, and something she pointed out about specifically UMAP and UMAP genetics and race, and I think Conrad brought it up a little bit earlier, is that it also doesn't represent, like, Conrad, you were talking about like, oh, you know, a family from Germany and a family from China, maybe like their kids meet and they get married, right? It doesn't represent people who come from different ancestries well. It'll kind of either try to put them in clump A or clump B, but it won't really represent this continuum of people that there are sort of between white people and Asian people, or white people and black people, or what have you. Like, it doesn't.
It's neither a real continuum, nor is it really a set of clusters.
It's a secret third thing.
Well, but it's not a secret third thing. No, when it comes to inheritance of genes, like we have like really, really wonderful models. We can fit these models. It's again, just these models will be much better at describing genes across people. It's just that that doesn't make really pretty figures.
So one quick question here. In the paper, right, the genetic ancestry in all of us, right? So what population did they use? Was it Americans or global?
It's Americans specifically. But they ask like self, so all the different like colors are coming from self-identified race. So they like fill out, basically like when you fill out a census form, it asks you like, are you white, black, Asian?
Yeah, I'm just wondering, I don't know, how would this graph look like if they did this in Brazil, where there's so much more miscegenation than in any other countries, right?
Right. I mean, the US, for all of the rhetoric about the US being this like great mixing pot or whatever, it's actually quite segregated. And I don't know that this would work in other countries and other contexts. And also, you know, there's the meta level of the race categories that we have are also arbitrary and are drawn and made up by people And they used to be different. And so if this had been done 50 years ago, there would have been different race categories that people could self-identify amongst. Yes, it's a social construct.
They were completely different. Every racialized society that's ever existed has had different categories of race based on whatever served those in power's uses of how they wanted to categorize people. And, just because the way the US does it would be very strange to someone, in a different place, at a different time.
I mean, even if you just showed it to people in Asia, they'd be like, wait, there's one category that's Asian? What?
Yeah.
Yeah, right.
That's true, right? Yeah.
Well, even when you look back at the colonial Spain and how many different categories there were and what those categories meant and all of that. And there's a little bit of overlap with that with North America, but even being so close to colonial Spain, we really don't have a system that is anything like that either. It's just totally foreign and separate because the power structures are different and the reasons that those classifications are important are completely different.
But yeah, the way that we do science is political. And it's telling to me that in the rest of this paper, which does a little bit of actual graphs that have statistics associated with them, there's only two more figures in this paper after the second one, but they kind of almost immediately forget about this whole grouping people based off of race or ethnicity. they go back to measuring just frequencies of specific genes that are related to diseases that they want to treat.
The purpose of all the figures is just showing how incredibly awesome their data set is. What they want to say is we recruited lots of people across race and ethnicity, which is a great thing. And the figure does the telling for them. That's why they did it. They don't want anyone to interpret the figure. They just want to say, look, there's a lot of red dots and a lot of blue dots and a lot of white dots too. And
but I guess if they wanted to do that, they could have just had a graph that said we had 100,000 white participants and 50,000 black participants and 25,000 Asian participants and
But it wouldn't have felt so scientific. Now, I want to just amplify Will's point here. The fact that these techniques are crazy complicated is not something that people avoid. It's something that they seek because it gives them a chance to say, well, this is really, really complicated, awesome science. And in reality, they're just hiding behind it because no one will say what that graph means. If you go that graph and you go to any of the authors of that paper, you're here's a graph. What does it mean? They'd be yeah, we just used UMAP. It's the paper doesn't say, here's the UMAP graph. This is what it means for you. This is what you now know about the universe, or at least about our population. It's more here's the UMAP graph. Isn't it pretty?
Interesting.
Yeah. I gotta say, although I think there was one graph, I think in the original UMAP paper, they did some graphs. I'm trying to look it up right now, where they just plot, I think, numbers by their prime divisors, and I would hang that on my wall. I gotta say, it's really pretty. It is beautiful, and it is a work of art, and absolutely meaningless, but whatever. I'll
okay, but it sounds like we agree, no?
I think so.
I mean, maybe not, but
yeah, the thing is, I didn't have the confidence that you have, Conrad, of saying this because I'm not a data scientist, right? I know that the mathematics is solid, mathematics as far as mathematics goes, right? But I don't have the expertise of how this is actually used in practice to say what's actually going on, right?
Yeah, I mean, look, my experience is that I've seen so many UMAP plots over my career, and I've never seen anyone really drive an argument from there. And usually the plots that we make are you say this about reality and I say that, and you're no, look at my graph here. Aspirin makes headache be less bad, and I'll be yeah, that graph is pretty convincing. I've never ever had that discussion with anyone in my career so far. And the problem is always people then I like, but couldn't it like here, like in, and at some level, it's yes, it could. And I don't doubt that it exists, but the vast majority of UMAP users is just a way of making pretty pictures.
And there's probably easier ways to make nice pictures as well.
Oh, I don't think so. No, like, let me argue that UMAP is like the ultimate in making pretty pictures, like
making abstract art.
The thing about like and so I don't want to disparage all of us too much, because I think that they're doing really valuable work and they have produced this great data set that now people can access and do their own, like basically subsets of research off of this huge data set that they've produced. But like, this was already an impressive undertaking, and it was already funded. Like, they couldn't have done it if it wasn't already funded to an extremely high degree. Their reporting on it was already going to end up in like Nature and also all over like science communication outlets, because it's already such a big undertaking. Like, I don't get why they had to hype themselves up more. Like, they already did it. The hype was already there.
So, but in that field, it's just a standard. Now, like, the problem with like practices like that isn't that it's their idea. No, like, it's not that like no one ever got UMAPs and then those guys were like, let's make our paper a little more attractive. It's rather that in that field, everyone does UMAP. Because as soon as you have a big data set, you can do UMAP. You can do UMAP again once you select for old people. You can do UMAP again with people with diseases. So like UMAP is kind of something that in that field everyone does. Therefore, it's almost like they were expected to run UMAP. Now, this paper is super visible. That's why we talk about this paper. The real problem isn't that this paper uses UMAP. That's not a problem at all. The problem is that countless papers use UMAP, and in countless papers, it doesn't mean anything, but it comes with like a false promise of meaning something. So if you're a new student, you go there, oh, like, look at that. And then someone is like, oh yeah, this is like really important. This is a UMAP. It's like really difficult math. And they'll be like starting to like read like papers from Will's community. And then they're like, but that has nothing to do with what I'm actually working on. And like, therefore, like all of a sudden, like a couple of months, they learn beautiful math instead of advancing cancer care or something. So,
but the thing is, I don't think that most people who are using UMAP are going to go look at how it works.
Yeah. And most of them don't. Like, most of them just use it to make a figure. And like everyone now makes that figure. It's kind of like it became a meme.
Right. I'm wondering if, in addition to your proposed disclaimer on all of the UMAPs about how they may not represent anything real about your data set, that like there needs to be education on, hey, you have this big giant data set. What actually would be what are useful ways to interpret it? Like clustering algorithms. Like, when do you, like, there needs to be education on when, on the scientist end, on like when you use which thing.
Jimmy, can I briefly just promote something here? There is a volunteer-driven large group that's called Neuromatch that tries to, at least within neuroscience and within climate change, teach students how they can use the various ones. Full disclaimer, I'm on their board, but like they exist in various domains. I'm sure the geneticists have the same thing, that they have courses where students go where they like learn how to do these things well.
Cool. Yeah. So I want to sort of wrap up by asking everyone like kind of what your takeaways were from this conversation, or like something interesting that you learned.
So my suspicions were confirmed, right? And one thing that I really liked, I don't know if we were recording already, but Conrad said, I don't know what question it's trying to answer, right? And I think that's my big takeaway. I think, which is something that we should ask more often, right? What is this? This is the answer to what question? I think I'm gonna even incorporate that into my algebra classes when I'm teaching, right? To sort of like get people to really think about what's going on and a bit more scientifically about things, and not just follow along with the trends, right? because I think one thing that I usually do a lot is, when I see a lot of conversations and discussions, I don't want to get like too much into like philosophy of science, but it's like, okay, so what is your prediction, right? What are you predicting here? Can you put your money where your mouth is, right? And UMAP doesn't give us that, right? It doesn't give us like something that we can then go on and test, right? This is what I feel like, which is really problematic about it,
Yeah, for sure. I think that question is probably the biggest takeaway for me from this, which is this is the answer to what question? And to like my imploring to all of the scientists that listen to this is to think about when you're using various different algorithms as there become more and more complex algorithms, is to think about like, what question does this answer for me and my data?
Yeah, for me, this is the big problem across probably much of science today, certainly in much of neuroscience. Like we learned by now that brains are really complicated. When we believed that brains were simple, our questions were simple. But now it's not that we replaced the simple questions with more complex questions. It often feels like we are losing the questions. And the reason why we have data is exactly to answer questions, like who will be healthy, who will have health problems, how does this part of the brain work or that part of the brain? And I think the data analysis methods we use, it's not about like, there can't be a magical silver bullet that like solves all problems. And like a data analysis technique is to answer a question. And I think like if there's one thing that I wish the whole world would know is that we don't analyze data to analyze data. We analyze data to answer a question. And as long as we're not clear what our question is, we're just guaranteed to mislead ourselves.
Yeah, absolutely.
I am just really excited that I am solidly on one side of a debate that none of my friends are going to know either side of it.
The best kind of debate.
Yeah, you guys have convinced me that, UMAP, if it ever comes up in casual conversation among my softball team or anything like that, I'll be able to say, you know what, I wouldn't trust it to make, to come to any sort of conclusion. It's a nice picture, and I will seem so smart and so well informed. So I will wait until that happens. Hopefully, the controversy goes on long enough to where I'll have that opportunity. So,
and this is already like a stone cold controversy. This is already so many months old.
I need to follow more people that Jamie follows on Twitter.
Jake is going to inexplicably start following science Twitter and be like, what the hell are you guys doing?
Good use of time.
An excellent use of time. Well, speaking of a good use of time, this podcast has been a great use of time. Thank you all so much for agreeing to record it at this hour of the night, and at least some of you with very little notice. So I appreciate you all for being on the podcast.
Awesome. Great. Thanks for having us.
Yeah.
Thank you. It was good to meet you guys.
Nice to meet you too, Jacob. And I absolutely love being here. It's great.
And once again, we've been talking about the paper Genomic Data in the All of Us Research Program by the All of Us Research Program Genomics Investigators. And we've been discussing this paper with our guests, William Lima, Conrad Cording, and Jacob Walker. You can find the paper and resources that we've discussed during this episode on our website at inplainenglishpod.org. Our logo is by the wonderful team at Biographics. That's Jessica Higginbotham, Tania Lintz, and Yolanda Campos. Our music is by Sam Brunwasser. You can find more of his work on SoundCloud at Visuals Now Beats. Finally, make sure to follow us on Twitter, Facebook, Blue Sky, and Instagram at Plain English Sci. That's PlainEnglishSci, so that you never miss an episode or updates about the podcast. Thanks once again for listening, and come back next time for another episode of In Plain English.